Good morning, everyone. At the outset, I would like to thank the organizers for inviting me to talk some of our research work that we have been doing at ISAR Mohali in this Complex Fluids 2021 conference. The title of my today's presentation is Differentiating Conformationally Distinct Alzheimer's A-beta oligomers using liquid crystals at aqueous interfaces. As you all know, the Alzheimer's disease is often characterized by the fibrillar deposits of A-beta peptides, as you can see in a brain MRI scan. The fibrillation of A-beta leads to beta amyloid plaque formation, which induces an oxidative stress and ultimately a neuron death. For a long time, it was thought that A-beta fibrils impart toxicity in the Alzheimer's pathology. However, the soluble oligomers of A-beta are the proximate toxins which vary in their structure and function. For example, hexamer, dodecamer, annular protofibrils, globulomer, and so on. They can bind to neurons, leading to memory impairments and toxicity and cell death, and finally Alzheimer's disease. It has been shown that soluble oligomers of A-beta correlate better with the disease progression, which can be classified based on their reactivity towards conformation-specific antibody. For example, you have two types of oligomer, let's say prefibrillar oligomers, which can recognize A11 antibody, that is anti-amyloid oligomer antibody. On the other hand, fibrillar oligomers can recognize OC antibody, that is anti-amyloid fibril antibody. This OC antibody recognizes the fibrillar oligomers with in-register parallel beta sheet structure, while the all antibody recognizes the prefibrillar oligomers with anti-parallel beta sheet structure. For example, if you have a monomer, it can go to a prefibrillar oligomer which is A11 positive and can have a conformation change leading to fibrils, which is OC positive. On the other hand, monomers can also go to fibrillar oligomer, which is OC positive and can act and add with a monomer, leading to fibrils that is also OC positive. Now, how does A-beta oligomers impart toxicity? The interaction of these oligomers with lipid membrane is considered to be a plausible mechanism by which the toxicity is imparted. The existing techniques employed to study protein aggregation require micromolar concentrations of the protein, which is far from the physiological relevance. Hence, there is a need for sensitive techniques which can report those binding events in concentration similar to the biological relevance. So what we need, an inexpensive and sensitive approach which is essential to study the behavior of A-beta oligomers on the lipid membrane, and we would like to know which of the two distinct assembly states is pathologically more significant, whether it is a fibrillar oligomer or prefibrillar oligomers. Now, here liquid crystal comes. Liquid crystals are the intermediate phases between the solid and liquid. 5Cb is the most widely used liquid crystal for sensing application, which is due to its safe anisotropy and birefringence that can give macroscale optical response towards small perturbations occurring at its interfaces. Although liquid crystal look like a liquid, but if you zoom it and see in the micrometer range, then we will see that mesogens within the liquid crystals that are near the interface, they can communicate their orientations to the mesogens that are a distance of 10 to 100 microns from the interface. Even short-range interactions between surface and mesogens can direct the ordering of bulk liquid crystalline phases. Now, in this situation, if the mesogens within the liquid crystals can experience some disturbance by targeted species, which can disturb the ordering at interfaces, it can extend throughout the distance, and as liquid crystal phases are optically anisotropic, it leads to different optical signals, and that can be easily visualized under a polarizing optical microscope. So we have been working on liquid crystals at ISAR Mohali. In particular, we are interested to see how liquid crystals can be used in biosensing applications. For that, what we need to create a stable LC aqueous interface which would be stabilized by capillary forces, where film thickness control is very important, and we can also use different microfabricated structures of various geometry to generate these capillary forces. So what you need to do? You need to create an interface between aqueous and liquid crystal. Liquid crystals can act as an oil, and this aqueous LC phase can get stabilized in different microfabricated geometry. For example, if you see a liquid crystal aqueous interface, what you can see a bright optical appearance under a polarizing optical microscope. Now, if you add different kinds of surfactants, lipids, proteins, they can couple to the ordering of the liquid crystals at these interfaces, and their presence and organization can be reported through changes in the optical appearance of the liquid crystal. For example, if you use a CTAB molecule, CTAB molecules can interact with the liquid crystal molecules at the interfaces, leading to a change in the orientation of the liquid crystal at those interfaces and leading to dark optical view. In 2002, Professor Abbott group showed that lipid adsorbed on the LC aqueous interfaces preserve the mobility in biomembrane. Subsequently, mounting reports showed that lipid-laden interfaces of liquid crystal can report lipid-protein binding events at nanomolar concentrations. So the questions we ask here, that can we distinguish between the two conformationally distinct A-beta oligomers by examining the dynamic spatial patterns of liquid crystals? Number two, which of the two distinct assembly states is pathologically more significant, whether it is pre-Farabilla oligomers or Farabilla oligomers, which is OC positive and A11 positive respectively. We started our exploration by preparing a mixed monolayer of POPC and POPG in 3:1 to mimic the anionic phospholipids in mammalian membrane. The liquid crystal films or monolayer films showed a dark optical appearance under a polarizing optical microscope, which is consistent with a uniform homeotropic alignment of the liquid crystal. The homeotropic alignment of the liquid crystal was stable up to a minimum of 24 hours. Before examining the dynamic optical response of the liquid crystal in presence of A-beta oligomer, we checked if the conformations of these oligomers is altered by the interfacial environment of liquid crystals, and for that we did a dot blot assay with OC antibody, which showed that the conformation of oligomers remain unchanged. Next, we see the dynamic optical response of lipid-laden liquid crystal films in presence of OC positive A-beta oligomers and A11 positive A-beta oligomers. The addition of conformationally distinct oligomers to the lipid-laden interface resulted in elongated birefringent domains. So to understand the lateral organization of lipids and proteins which resulted in the pattern orientation of liquid crystals, as you can see in the previous slide, we used rhodamine DHP doped lipid monolayer. We see that dark regions of liquid crystals are red fluorescent, while the bright domains in the cross-polarized microscopes appear dark. This suggests that the non-fluorescent bright domains are lipid linear. To further understand these branch domains, we performed a THT assay. THT is a well-known amyloid marker which binds to the fibrils to give bright green fluorescence. We found that branch domains are THT positive amyloid fibrils. We further performed atomic force microscopy imaging of the interface, which showed curved amyloid fibrils of height 10 to 16 nanometer. We have also performed control AFM experiments of the oligomers in solution, and just after adding on to the interface, we found oligomers of various sizes, but there are no fibril formations. After establishing that the oligomers interact with the lipid monolayer and mature into amyloid fibrils, we aimed to see the kinetics of the fibrillation. We measured the grayscale intensity of the polarized optical micrographs captured every 5 minutes and fitted the data to a single exponential equation which gave us the T-half values. The OC positive A beta oligomers exhibit 9 times faster fibrillation rate in comparison to A11 positive A beta oligomers. Next, we looked spatiotemporal evolution of these domains and found that by varying the concentrations of the oligomers, the different patterns comes. For example, if you have the OC positive A beta oligomers, it can be sensitive at a concentration of 35 nanomolar. On the other hand, below 35 nanomolar, they are unresponsive. In case of A11 positive A beta oligomers, we see the formation of branch domains at a concentration of 50 nanomolar. Below 50 nanomolar, there is no formation of elongated domains. In order to compare the dynamics of LC reorientation in presence of both the oligomers, we plotted the grayscale intensity of the optical micrographs in contact with different concentrations of the oligomers and found that the lipid-laden liquid crystal is more sensitive to the OC positive oligomers. So this is the model which depicting the interaction between lipid monolayer and A beta oligomers. So what we can see, the A11 positive A beta oligomers have lower sensitivity. On the other hand, OC positive A beta oligomers have faster sensitivity and fast formation of fibrils, and therefore they can bypass the toxicity. On the other hand, A11 positive A beta oligomers can remain in solution in oligomeric form, leading to more toxic compared to OC positive A beta oligomers. So in conclusion, what we found, the dynamic response of liquid crystal was different for the two conformationally distinct A beta oligomers. The OC positive A beta oligomers show a 9 times faster fibrillation rate in comparison to A11 positive A beta oligomers. The detection limit of OC positive A beta oligomers was found to be 35 nanomolar. No apparent change in the optical appearance of liquid crystal was observed for A11 positive A beta oligomers at 35 nanomolar. We also see faster fibrillation of OC positive A beta oligomers, which might help in bypassing toxicity. The slow aggregation of A11 positive prefibrillar A beta oligomers might prolong their existence in the oligomeric state, thus making it more cytotoxic species. I would like to thank my student, Ipsita, Professor Samrat Mukhopadhyay and his group, his student, Ms. Priyanka Madhu, and I thank all the funding agency, DST, DAE, INSA, CSIR, STARS, ACRB, and of course ISAR Mohali for generous funding support. Thank you very much for your kind attention.